What if I told you that life can begin in a laboratory? Every year, millions of babies are born not from the usual process, but from a procedure done in a glass dish by scientists in white coats. This is IVF, and today we're breaking down exactly how it works step by step in plain English. Test tube baby is actually a nickname. The real term is in vitro fertilization, which simply means fertilization outside the body. In vitro is Latin for in glass. So yes, the name is almost accurate, except it's a dish, not a test tube. The first test tube baby in history was Louise Brown, born in 1978 in England. Today, over 12 million people have been born through this method worldwide. But how does it actually happen? Let's walk through it. Normally, a woman's body releases one egg per month. But for IVF, doctors need more than one to increase the chances of success. So the woman is given hormone injections for about 10 to 14 days. These hormones tell her ovaries to produce more eggs than usual. Doctors monitor the process closely using ultrasound scans, watching the egg follicles grow in real time. Think of it like this. If you're a chemist trying to run a reaction, you don't use just one drop of reagent. You prepare enough to work with. Once the eggs are mature, they're collected in a minor procedure called egg retrieval, also known as follicular aspiration. The doctor inserts a thin needle through the vaginal wall, guided by ultrasound, and gently suctions the eggs out of the follicles. This takes about 20 to 30 minutes. The woman is sedated, so she feels little to nothing. The eggs are then handed to an embryologist, the scientist in the lab, who examines them under a microscope and selects the healthiest ones. Now here's where the real science happens. The eggs are placed in a special culture dish filled with nutrients that mimic the inside of the human body. Then sperm is added. In standard IVF, the sperm and egg are left together and fertilization happens naturally in the dish. But sometimes, if sperm quality is low, scientists use a technique called ICSI, intracytoplasmic sperm injection. They pick a single healthy sperm using a microscopic needle and inject it directly into the egg. Imagine using a micropipette to deliver one precise molecule into a cell. That's basically what's happening, just on a human scale. After fertilization, the egg becomes an embryo and is kept in a carefully controlled incubator at the same temperature as the human body, 37 degrees Celsius. Scientists watch the embryo develop over 3 to 5 days. By day 5, it reaches a stage called a blastocyst, a small cluster of about 200 cells with a fluid-filled center. This is the stage where the embryo is strongest and most likely to implant successfully. The healthiest embryo is selected, and the others can be frozen for future use. The selected embryo is loaded into a thin, flexible catheter and gently placed into the woman's uterus. There's no surgery and no incisions. It's a quick and mostly painless procedure. After this, the woman waits about 10 to 14 days and then takes a pregnancy test. If the embryo implants into the uterine wall successfully, a baby is on the way. The success rate varies, typically between 30 and 50% per cycle, depending on age and health factors. So that's IVF, one of the greatest intersections of medicine, chemistry, and biology ever developed by science. From hormone injections to microscopic fertilization to a growing embryo, all of it works together to give families a chance at life. If you found this fascinating, drop a comment below and let me know what medical science topic you want us to break down next. And don't forget to like and subscribe. We make complex science simple every week.